Hi everyone, we're looking at chapter 12, which is all about this famous scientist named Johann Gregor Mendel, and what some of his experiments on pea plants taught us about inheritance patterns. So to start, we have to kind of parse out the difference between dominant and recessive phenotypes. Recall that a phenotype is basically the way that one would appear. So my hair color, for example, is what you call the phenotype, or the shape of my nose is a phenotype, or how tall I am is a phenotype. So these things are called phenotypes are basically the appearance of what are known as traits, or traits are basically different attributes that an organism might have. And what Mendel discovered is that there are two different types of traits. There are dominant traits and recessive traits. And how it works is that if somebody has a copy of a gene that's a dominant trait and a copy of a gene that's a recessive trait, well, the one that's called dominant will be the one that is shown. That is to say, that person would have that phenotype. And then on the flip-flop, if somebody only had recessive genes, then they would have the recessive phenotype. So kind of here's an example. There's a whole bunch of traits out there, and this is kind of fun. Tongue rolling. Can you roll your tongue? Stick your tongue out and make it kind of in a rolled shape. If you can, that's the dominant trait. If you cannot, that's the recessive trait. And so let's say, for example, your dad gave you a tongue rolling gene that allowed tongue rolling, and your mom gave you a gene that was recessive. There was no tongue rolling ability. Well, the dominant one is the one that's always possessed or expressed by the individual. So you inherit a non-rolling tongue rolling gene from your mom and a tongue rolling gene from your dad. Because the dominant one is tongue rolling, you will be able to roll your tongue. Earlobe attachment. If you have ears that have free earlobes, so if this was a picture of an ear, you have an earlobe that actually hangs down like this, versus an ear where the ear is just going to come straight to the side of the to your head. All right, so this would be free. If your earlobe is like that, that's dominant. If you have one that's attached, that is the recessive trait. So one thing that's interesting about recessive traits is that it requires that you receive that trait, the gene for that trait, from both your mom and your dad. So if you have attached ears, I would guess that both your mom and your dad have attached ears. Whereas if you had free earlobes, then it could be the case that just your dad had free earlobes, but maybe your mom had attached ears. You just need one copy of the dominant gene to have that phenotype or that appearance. Another one is pinky shape. If you extend your pinky out and it's bent, or it kind of bends off to the side, that's the dominant. And then, whereas if it's just totally straight, that would be recessive. Arm folding. If you go ahead and cross your arms right now, do it in just the way that's most natural. Go ahead and fold your arms, cross your arms. If you look in the right is on top, then that's dominant. If the left is on top, that is recessive. A cheek dimple. Do you have dimples on your cheek? If you do, that's the dominant. And if you don't, that's recessive. A cleft on the chin. Is there like a little dimple on your chin? If there is, that's dominant. If not, it's recessive. Hitchhiker's thumb. If you stick your thumb out like a hitchhiker, is it straight up and down or is it hooked? Those are the dominant and recessive there. And then toe length. Is the second toe longer or is the first toe longer? The first toe longer would be recessive, and again, that would mean that you probably, both your mom and your dad gave you that, because the only way you can have a recessive trait is if both the traits that you inherited from your parents are recessive. As soon as you inherit just one dominant, either from mom or dad, you will have the dominant phenotype. Widow's peak. If you look at hairline, and there's like a little kind of indentation there on the hairline. So these would be the eyes. Here's the hairline here. That would be called a widow's peak, and that's the dominant trait. And if vice versa, the hair is just straight across, that's the recessive. And then one last one. Can you see the number that is shown in this picture? This is a little bit more of a rare one, and oftentimes I don't have any students that can't see this, but occasionally I'll get one. If you can't see the number, that is the trait of red red-green color blindness, an inability to distinguish the difference between kind of similar shades here of red and green. And somebody that is red-green colorblind could not see that there is the number embedded in here, 74. This is the recessive trait. So those are dominant and recessive traits. And that gets us to what are known as alleles. Allele is basically just basically the variations of a trait. And so generally there are two alleles for every trait. One's dominant and one's recessive, just like we talked about. So specifically though, alleles generally refer to individual chromosomes. Okay. So we're kind of connecting the idea of one's appearance to the codes that are on their DNA. And just keep in mind throughout all of this, it's the appearance, the phenotype of an organism that is determined by its genotype. So the genes always determine the phenotype. Okay? So what I've done here is I've included a karyograph here of a bunch of different chromosomes. So this would be kind of like a human karyograph, because you can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23 pairs of chromosomes for a total of 46 chromosomes. So that would be the same as the cells that we have. And on each one of these chromosomes, these are homologous chromosomes now. Anytime you see them paired like this, chromosomes of the same shape and bearing the same the information for the same genes are called homologous chromosomes. And each one of these pairs, one of them came from dad and one of them came from mom. Okay? One of them was brought forth by the sperm and the other one was harbored by the egg, and when the sperm and the egg met, it formed a zygote which had two sets. And then from that zygote, that identical copies of cells were made until you had a whole organism. Okay? So on each one of these chromosomes, there's going to be different traits. Like, so for example, I don't know exactly which chromosome it is, but hair color, let's say. Maybe it might be that the gene that codes for the pigment that determines what your hair color is, could be that the information is stored, let's say, on chromosome 3, right at this spot of the chromosome. So right in here, there are millions and millions and millions of base pairs of DNA which contain the information your body needs to build the hair in a way that has a certain color. Okay. Well, generally homologous chromosomes, the way they work is that they're going to have the same genes on the same, what's called locus. So this, where this is on a chromosome, is called a locus. So right here, this would be a gene that determines hair color. Well, in that same spot on the homologous chromosome is also a gene that controls hair color. And this one would be, let's say, the one that came from mom, and then this one would be the one that came from dad. So let's say that dad, in the process of meiosis, when sperm was being produced, it packaged up a chromosome that bears the gene for blonde hair color. Okay, so it could be that your dad had at least, maybe he didn't even have blonde color, but in his genome he had a gene for blonde hair color, and when it was packaged up into a sperm and it met this egg, it led to this combination. So there's one chromosome from dad that's got blonde hair color, and let's say your mom put forth brown hair color. So now they're in this one cell, and this again would represent, if you took any cell in your body that's not a gamete, it would represent this type of a diploid cell where it's got two copies of every gene. So in this particular instance, this cell would have two chromosome number 3s. One would have a copy for the information required to make blonde hair. The other one would have a copy for the information required to make brown hair. And then the question is, well, what is this organism going to have? Is it going to have brown hair? Is it going to have blonde hair? And that's where we get into this whole idea of what the difference is between dominant and recessive alleles. There's the brown allele present, or there's the blonde allele present, and there's the brown allele present. And we might say that the dominant one, in this case, for hair color, the dominant one is the brown color. Recessive is blonde. So this individual, if this is one of their cells, they in their DNA, which is the same for every single cell, they have one copy of a gene for brown hair, one copy of a gene for blonde hair, one copy of a gene for brown hair. But because brown is the dominant one, this is going to be the gene that is expressed. So this is going to be the one that's made active, and then everything's going to be made to create brown hair. Whereas this one over here is just going to kind of remain silent. It's not going to really do anything. Okay, so that's kind of how this works. If there's two traits on these homologous pairs, the one that's dominant is the one that does the stuff, and then the other one just kind of remains quiet. It's kind of just a backup. And for whatever reason, that's just how it works. You have two copies of every gene, one on either homologous chromosome. Okay, then this gets us to this idea known as an allele key. So generally in all of these things, one wants to make an allele key. And so our allele key could look something like this. We might represent that B stands for a brown allele, and maybe lowercase b stands for a blonde hair allele. And then from this point on, what this means, and when we talk about these keys, it's actually talking about the individual chromosomes in an easy way. So if this was the one that had brown hair, we would say that this is a big B, and if this was the one that had blonde hair, we would say that it's a little b. So we can go on then to talk about one's genotype just using this allele key for the specific trait. So if this was the case and this individual had one copy of the blonde hair and one copy of the brown hair, we would say that they are BB. Okay, they're big B, little b, meaning they have one copy of the brown hair gene, one copy of the blonde hair gene. We're not specifying which one of these genes came from mom and which ones came from dad. That's not really all that important. What is important, at least in this chapter, is just to know that we have two copies. It could be the case, though, that perhaps maybe that both of your parents donated a brown-haired gene, a brown-haired trait, and then we would say that this individual was big B, big B. Or on the very last option here, it could be that both mama and dad just brought out or produced a little b and a little b. So this chromosome which came from the sperm and this chromosome that came from the egg, when the process of meiosis occurred, this sperm got the little b and this egg got the little b. Okay, so you could be big B, big B, big B, little b, or little b, little b. Those are kind of the combinations for any one of these traits when we're talking about just one trait and there only being two alleles, a dominant allele and a recessive allele. So this gets us to these words called homozygous versus heterozygous. If one has two copies of a allele, we would say it's homozygous. This word homo generally means the same. Okay, so homozygous would mean the type of genetics that is the same, so they have the same type of allele. Heterozygous is this one right here, where they have one copy of the dominant and one copy of the recessive. If it was homozygous and the two alleles were dominant, we would say that's homozygous dominant. If it was homozygous and the two copies were recessive, we would say it's homozygous recessive. And then if one has just a dominant and recessive allele in their genome, we would say it's heterozygous. And that's kind of how it works. And what can happen, at least according to the modern theory of evolution and survival of the fittest, is that various mutations can occur which can produce new alleles. Okay, so there might be a variety of alleles that are produced. Maybe just instead of brown and blonde, suddenly there's a slight bit of red hair that's introduced. Red hair is actually, I think, a mutation that occurs on chromosome number 4. So actually, perhaps some of these hair color things are on chromosome number 4. Or there could be a mutation which creates kind of like a white hair, or even like maybe someday there'll be a mutation that creates kind of a bluish hair. So that's how a little bit about how variation in traits and things can introduce is oftentimes a mutation is kind of the culprit of creating that trait. All right, so a little bit about what alleles are, what the difference is between dominant and recessive, and then what these two words mean, homozygous versus heterozygous. But in all of these things, just keep in mind the nature of a diploid nucleus. Remember, we've got two sets of chromosomes, so because of that, for every gene, there's going to be two copies. They might be the same type of copy in terms of the same type of trait, or they might be two different alleles. Okay, and the key there is this word allele. All right, so how does this guy Gregor Mendel fit into the picture? Well, a little bit about Johann Gregor Mendel. He's oftentimes considered what's known as the father of genetics. And that's because he really pioneered some studies that gave us the foundation in the framework for understanding how things are inherited. So here's a picture of Gregor Mendel. He would live to be about 62 years old, and he was in kind of the mid 1800s. And what's kind of interesting about him is he was actually a priest. And so he was a priest, but also he was a teacher, and he loved to learn, and he was also clearly a scientist, and then, as we said, a priest, so a man of faith. And in those days, Gregor Mendel grew up on a farm, and I think he was just tired of farming, but they didn't quite have the money to go to school. So in those days, if you wanted to get an education and go to school, you could commit your life to following the faith and being a priest, and then education was paid for. So that is kind of the route he took. And I feel bad for Gregor Mendel. Gregor Mendel had some really interesting ideas that went completely unnoticed. In fact, nobody even really paid any attention to his ideas because they didn't really quite understand what he was trying to say. And so he died thinking probably that his life wasn't a very significant contribution to the field of genetics. And it was only about 40 years after his death that people started to discover his work. And even further than that, Gregor Mendel wanted to be a high school teacher. And so there was a couple of times he took some exams to be a high school teacher. And he got all the way through the exam, but then in the very last part, the oral part, he failed it. And so he failed it once and couldn't be a teacher. And then 10 years later, he tried again and he failed it again and couldn't be a teacher. And so I think Gregor Mendel had kind of struggled with a little bit of depression. And you can imagine being someone with such a brilliant mind that doesn't really get any credit, and furthermore is having a hard time pursuing a career. That would be a tough thing. And me being somebody that's failed several big exams like that, I can know that's a little bit of a frustrating process. So give Gregor Mendel some credit now, because he sure deserves it. Okay, but anyways, what he did, he conducted a decade-long experiment on pea plants, and to determine their different inheritance patterns. And what he found was something that countered this idea of what's known as continuous variation. In this day, the day of Gregor Mendel, they thought that offspring would inherit a trait that was kind of a blend between the two parents. So, for example, if my dad had really, really, really brown hair and then my mom had really, really, really blonde hair, I would inherit hair that's like somewhere kind of in the middle or something. Okay, and it's hard to imagine how they would have thought that, because clearly, you know, there were kids out there that had hair that was blonde when both of their parents had brown hair, or vice versa. So it's kind of curious why they maybe thought this. But anyways, that was what we mean by continuous variation, that there's like a spectrum of variation, and what an offspring gets is some kind of a hybridization or a merging of the two traits of the parent. And he's saying, no, the traits are discrete little units where you either get it or you don't get it. And that was a pretty new idea. And this is how he came about arriving at those conclusions. What he did is he grew up a bunch of pea plants, all right? And these were called true breeding pea plants because generation after generation after generation, he would always select the violet ones and then the white ones and keep them separate, and let them mate separately for generations until they would only breed violet flowers, and then the other population would only breed white flowers. So there's a bunch of plants over here. They are only white flowered pea plants, and then there's a bunch of plants over here that are kept separate, and there are always only purple plants, and they only produce purple flowers, and these ones only produce white flowers. Once establishing those, which are called true breeding, okay, for true breeding, they don't change the color in their flowers after generations. After he achieved those populations, he would take some of the pea plants with violet flowers and have them crossed with some of the pea plants with white flowers. And the first curious thing that he found is when he did this, The entire next generation, which we would call the F1 generation, they had only purple flowers, which is interesting. According to what the people were arguing about in those days, it should be the case that a purple flowered plant and a white flowered plant mate to produce a flower that's like kind of a color in between purple and white, so maybe like kind of a more of a pinkish rosy kind of color. But he showed that it was all or nothing. It either got purple or white, and in the first offspring, the first generation was 100% purple. They were all purple just like these ones. Not even a hint of white anywhere to be found. So that was curious, but then what's even more curious is he would take all of these offspring and he would cross them with one another, and then all of a sudden the white flowers would come back into existence. And that was really bizarre. And furthermore, when he did this, he would find that the ratio between these two alleles was usually about 3 to 1. So in this experiment, he counted 705 violet flowers to 224 white flowers. And this was consistent through a variety of traits that he looked at. He looked at flower color, as I mentioned, and then also flower position, plant height, seed texture, seed color, pea pod texture, pea pod color. For all these traits, when he counted up all of the F2 offspring, so that second generation, it was always, the traits of that generation were always about 3 to 1, 3 to 1, close to 3 to 1, 3 to 1, 3 to 1. And that was just so consistent, it was hard for it to not be coincidental. Okay, so in retrospect now, when we look back at this, I want you to consider what's going on. A true breeding, well, let's make an allele key, I guess, first. Anytime you're thinking about these kind of genetics things, it's good to make an allele key. The two alleles, or the two traits out there, are violet and white flowers. And which one do you think is the dominant and which one do you think is the recessive? Well, I'll tell you that the violet flowers is the dominant one. And the way that I know that is because the dominant one would be the one that's present here in the F1 generation. Okay? And then the white flowers, these are, this is the recessive allele. Dominant allele. So our allele key will say that violet flowers, let's say that the violet flowers will denote an F here for flower. So a capital F always stands for the dominant, a lowercase f always stands for recessive. So we'll say that a capital F is for violet and a lower f is for white, okay, because those are my two alleles. Well, you can imagine if these are diploid plants, well, this plant here is going to have 2 copies of the gene, and if it's true breeding, that means it's homozygous, okay? Meaning there cannot be any introduction of a new allele into the population if they're breeding with one another. So these ones are homozygous dominant, so they would be big F, and these ones here are homozygous recessive, so that would be little f, meaning these plants, when you look at their diploid genome, whatever chromosome it is that determines white flower color, both the chromosomes in the homologous pair each bear the recessive trait, the information that codes the recessive trait. And if you look at the 2 chromosomes of the homologous pair in this genome, each of the chromosomes on the homologous pair of whatever chromosome it is that determines flower color, each one is going to have a dominant information that codes for the dominant allele. Okay. Well, when you cross these together, every single bit of offspring gets a genome that's like this: big F, little f. They inherit a little f from this plant, and they inherit a big F from this plant. And so the diploid genome here of these plants is big F, little f, meaning they have one copy of the dominant allele and one copy of the recessive allele. And you can see that even though they have a copy of the allele for white flowers, because they have that dominant allele as well, it's always the dominant one that is going to be the phenotype that's visible. Okay, so that is why their actual phenotype, even though their genome has a copy of each, their actual phenotype is purple because they have just one dominant allele, and if you have just one dominant allele, you will have that trait. Okay, and then he had all of these plants that had one copy of the white allele and one copy of the purple allele, and then when he crossed those together, he got this. And why this ratio is the way that this is gets us to actually a peer discovered. His name was Punnett, so we call it a Punnett square, is the tool that can be used to explain this ratio and also predict what ratio we might get in any other type of scenario involving one trait. All right, so the Punnett square, good segue, is a tool that we can use to predict what the phenotypes of an offspring will be if we know what the genotypes of the parents are. And there's a couple different ones. There's ones for what's called a monohybrid cross. That would mean just one trait, or it can be used for one trait. And then a dihybrid cross, these can be used for two traits that are inherited together. Kind of, we usually think about inheriting and being inherited at the same time. And then X-linked traits. X-linked traits are things that are found in the X chromosome, which can be a little bit different for a reason that I'm going to explain in just a minute. The first, the monohybrid cross, okay, we'll think about it again. Let's say we'll do some sort of trait here. And let's say the trait is the widow's peak. Okay, so the trait is the hairline, like we mentioned earlier. And there's going to be a dominant allele and a recessive allele. And the dominant allele in this scenario is to have a widow's peak. And a recessive allele is to have a straight hairline. Okay, so keeping that in mind, let's make a quick allele key here because we're going to need to keep track of traits that are being inherited. So the allele key would look something like this. Let's designate, we're talking about hair color or hairline, so let's just use the H for our key. So we'll make the first one capital because the capital one is always referring to dominant, and we'll say that is a widow's peak. And then a lowercase h we'll say is a straight hairline. Okay, so then the question is, let's take a look. All right, so let's say that mom is heterozygous. And let's say that whatever genetic information codes for this trait, the hairline, let's say maybe that's found on, I don't know, chromosome 11. I don't know where it's actually found, but let's just say it's on chromosome 11. Well, mom has two copies of this trait, and let's say for this example that mom is homozygous recessive, meaning that she has a straight hairline, and because she has a straight hairline, it's because one of her chromosome number 11s has the recessive allele, and the other chromosome number 11 that she got has also the straight allele. The only way that one could have the recessive phenotype is to be homozygous recessive, remember. And then let's say, so mom has got a straight hairline. Then let's say dad has a nice widow's peak. And let's say that dad is heterozygous. Well, if dad is heterozygous, that means that one of the chromosomes he inherited from his parents bears the dominant allele, and another one of the chromosomes that he inherited from his other parent is going to have the recessive allele. Okay. Now the question would be, if mom, who has straight hair and has a homozygous recessive genotype, and dad, who has a widow's peak and is heterozygous genotype, the question would be, if mom and dad make babies, what percentage of their babies are going to have, let's say, a widow's peak versus a straight hairline? So a Punnett square is really going to answer the question, what is going to be the different probabilities for the different genotypes of the offspring? Okay, so the way that this is set up, they call it a Punnett square because you really just have to make a square. And it's pretty simple, but sometimes it can be kind of tricky to keep in mind what it is that we're actually keeping track of. So here's the square. Okay. How a Punnett square is set up is it looks like this. There's 4 squares when we're talking about one trait. And along the top, you will put the genes, or maybe we'll keep it language similar here, the traits that could be provided by one gamete. So let's say on the top here we're talking about dad's gamete, so that would be sperm. And then on the side here we'll list the different genes or traits that could be provided by another gamete, or probably by the other gamete. And so we'll say that the other one then would have to be the egg, right? So here's where we have a little throwback to the previous chapter that we were talking about, meiosis. This is what dad's nucleus looks like in all of his somatic cells, his body cells. Well, recall when meiosis happens and dad is producing sperm, what's going on is this diploid cell is breaking down into 2 haploid cells, or ultimately 4 haploid cells for every one starting cell. So all that to say, you know, when sperm are being produced by dad, here's the little sperm, here they are, that sperm can either inherit an H, or it can inherit a big H, or it can inherit a small h. It all depends which side these chromosomes lined up on when it's in metaphase 1, because that's going to determine, you know, where it goes where. So there are 2 combinations of sperm that dad is producing. When he's producing millions of sperm each day, about half of them are going to have carry this gene with them. The other half is going to carry this gene. So here are the different combinations. You could have one that's carrying the big H, or one that's carrying the little h. Okay, and that would be the same for all the different traits out there. There's going to be, for any of these chromosomes, there's 2 combinations. The sperm can either carry this one, or it can carry this one. And so you can start to see when that's the case for every homologous pair, there's really an infinite number of variations that one sperm could be. You know, you could have one sperm that has this one, this one, this one, this one, this one, but then maybe over this one over here for chromosome number 6, and then the rest of them has this one here, and so on and so forth. You get the idea that there's a lot of different combinations there. Okay, so then over on this side, when we think about the different gametes, the different traits that could be passed on by mom's gametes, mom has already made all of her gametes prior to even being born, believe it or not. So that's a little bit different than men. But in either case, the egg that she could be providing could either have this copy of the allele, or it could have this copy of the allele. And since they're both the same, it doesn't really matter. We'll just put H and H. Okay, so these are the different eggs that could be produced by mom regarding this specific trait. So when you look at the different combinations of gametes that dad could produce and the different combinations of gametes that mom could produce regarding this specific trait, then we can go through and kind of take a look at what would happen if each one met. So if this sperm from dad met this egg, then that zygote would be big H, little h, because it would inherit this trait from the sperm and it would inherit this trait from the egg. If this sperm was to meet this egg, then that would be a little h, because it would get this copy, the recessive allele from the sperm and the recessive allele from the egg. If this sperm was to meet this egg, it would be big H, little h again. And then if this sperm met this egg, it would be little h, little h. So these are all the different combinations that could happen in terms of the fertilization event. And so when you think about this, you can take a look. 2 of the 4 possibilities would lead to children who have are homozygous recessive, so that would mean that they have the straight hair line. Okay, and then 2 of the 4 over here have the dominant allele, so these would be the widow's peak. So if I was interested to know what the phenotypes would be of the offspring concerning this trait of hairline, and I had this scenario with these 2 parents, I could probably bet that if mom and dad had like a super family and they had like, let's say like 6 kids, I could probably say it's safe to say that about 3 of those kids would most likely have straight hair, and 3 of them would have the widow's peak, just because I know, given these probabilities, there's about a half, 50% probability that it will have a widow's peak or a straight peak. It's kind of 50-50 based upon these combinations. Okay, and this is going to vary depending on if we're talking about mom being homozygous or heterozygous, or dad being homozygous or heterozygous for a trait. And so in lieu of some of this information, I want you to see if you can think back about why we're getting this 3-to-1 ratio here in Mendel's experiments. So what's happening here, if we do a Punnett square, just to keep this in mind, and let's say that we're talking about a big F, we'll make our allele key here for the Punnett square, big F equals a purple flower, and little f equals a white flower. All right, these are true breeding here in the first generation, so that cross looks something like this. One parent, the 2 different traits that it could pass off in its gametes would be big F, big F. Okay, that would be the gametes that this plant could make, and this plant could bring donate the different traits of little f in its gametes. And so these are the only combinations of eggs that can be made, and these are the only combination of sperm that can be made from one plant, and vice versa. So, when these gametes meet, you can see that because of the setup, every single combination of these gametes meeting results in a zygote that is heterozygous. Okay, so that's all of these plants have the genotype big F, little f, meaning they bear 1 copy of the purple flower allele and 1 copy of the white flower allele. Well then, if we cross all these plants together, the different gametes that could be brought to the table here are big F and little f for each plant. So, plant number 1 could produce a sperm that either has the big F allele or it has the little f allele, and plant number 2 could either have an egg that has the dominant allele or the recessive allele. Well, if you cross these 2 together, then here are all of the combinations that could happen. And you can see that in 3 of the 4 combinations, there is a dominant allele inherited. And so that is going to result with a dominant phenotype, which is the purple flowers. And only 1 out of those 4 is going to be the recessive. So, given the probability of all of this, you can see there's a 3-to-1 ratio, dominant to recessive phenotype, and that's exactly what Mendel found in his studies. Okay, so just through counting some flowers of pea plants, he was able to kind of come up with some pretty hefty stuff. I mean, talk about awesome scientist, right? Okay, there's one issue though. Let's say you look at a plant with a dominant phenotype, and you're asked the question, is this homozygous or is this heterozygous? This could be, keeping in mind our same allele key, this could be a big F, little f, or it could be a big F, big F. In either case, it's going to be the same phenotype. We don't know just by looking at it if it's heterozygous or homozygous dominant. Well, one thing that can be done is if you take this type of plant, you can do what's called a test cross and breed it with some true breeding recessive plants. And the idea is that if you were to cross this with a true breeding recessive plant, you can see that if these 2 were crossed together, it could be the chance that this gamete gets donated and this gamete gets donated to produce offspring that are FF, which would be a white flower, because that's homozygous recessive. So as I say, if you cross any plant that's a dominant phenotype with something that has the recessive phenotype, you can kind of determine if the original plant you're wondering about is heterozygous or homozygous, because if you did that cross, then if there are any recessive phenotypes that come out of that the offspring, then you know the original one was heterozygous. Whereas if you did this cross here, every single one of the offspring would be big F, little f, and that would be a dominant phenotype. So you would not expect there to be any white flowers from the offspring of crossing this with the recessive. So just kind of a picture here. Let's say you have, there's a dominant allele or a dominant phenotype, but you're not sure if it's 2 copies of the dominant allele or just 1. Well, cross it with a true breeding, one with the recessive allele, because that one's bound to be homozygous recessive, and then you just look for the offspring. You can see if this one here was a big Y, then we would get all of these offspring, and you can see that none of them are the recessive phenotype. Whereas if this unknown one was a little y, then when you do the Punnett square, you actually get about half of them having a recessive phenotype. So that's a kind of a sneaky way to do a little kind of genetics just by looking at the phenotype. So that's a monohybrid cross. Now a dihybrid cross is like taking it up a couple notches here on the complexity scale, because now we're dealing with 2 traits. So the way that this would be done, let's say that there are 2 traits. Trait number 1, let's say, is flower color. Just keep it consistent. And let's say that we are also interested in another trait, trait number 2, which let's say is seed texture. Well, the dominant allele in flower color we've already talked about is purple, and the recessive allele is going to be white flowers. And in seed texture, I believe, oh my gosh, I'm blanking. I believe it's smooth. Dominant, I believe, is the smooth phenotype. And recessive is wrinkled. Okay, so let's put our allele key up. We'll say that big F equals purple, little f equals white flowers, and then that's for trait number 1. And then let's say for trait number 2, let's say that big, talking about texture, so let's say that big T equals smooth, and little t equals wrinkled, since that's the recessive phenotype. Okay, all right, so here's our allele key for 2 separate traits. Now let's see how this might play out in a Punnett square. Anytime a dihybrid cross is done, it requires that a Punnett square be made that has 4 columns and 4 rows. So we'll go ahead and make that, and you'll hopefully see why that is in just a moment. So here is a nice Punnett square, and here is 1, 2, 3 columns, and here is 1, 2, 3, 4 rows. And so now let's consider a couple of things here. Let's consider plant A. Oops, plant A, and then we also have plant B. And let's say that plant A has the genotype big F, big T, little t, and let's say that plant B has, oh gosh, what do we want to do here? Let's say that plant B has little f, big T, little t. So when you look at these two traits for flower color and texture of the seed, plant A has, when you look at its diploid genome, one of its homologous chromosomes bears the dominant allele, one bears the recessive allele for flower color, and that's the same for texture as well. And then when you look at plant B, look at the homologous pair that bears the info for the trait of flower color. Both of the chromosomes in the homologous pair have information that codes for the recessive allele, but then they're heterozygous for texture. All right. So the question then is, what kind of gamete possibilities could we produce here with plant A? And what kind of gametes could we make with plant B? And so there's a couple different combinations. When this plant A goes through meiosis and the gamete is being produced and these genes are being packaged into it, you could have one gamete that takes this allele and this allele and puts it into the gamete. So that would be a big F and a big T in that gamete. Or you could have one that takes this big F and packages it with the little t. So that would be a big F and a little t. So I guess that kind of assumes that these two traits are on different chromosomes, not the same chromosome, because that's the only way that they could kind of be packaged up into different bits. Well, that gets actually a little bit more complex, so I won't even go there. Okay, so that's the first two combinations, and then the other combination is that this allele can be packaged up with this allele, so that would be a little f big T, or it could be that this little allele is packaged with this little allele, so that would be a little f little t. So those are all the gametes that could be produced from that plant. And then here, this one could be packaged up with this one, so that would be a little f big T. Or this one could be packaged up with this one, so that would be a little f little t. Or this one could be packaged up with this one, so that would be a little f big T. Or the second one on each would be little f and little t for all the gametes that they can make. Okay, so now you got quite a few possibilities. It could be that this gamete meets this gamete. If that was the case, it'd be big F, little f, big T, big T. Could be that this gamete meets this gamete. And again, if it's helpful for you to conceptualize what's going on here, just think about these all being sperm and these all being eggs. Okay. So it could be that the sperm that bears these two alleles meets the egg that bears these two alleles. Or this sperm that has these two alleles could bear this one, and that would be the same result, big F, big little f, big T, little t, and that's going to be, over here this is two little f's, so we'll go little f, and then two big T's, so big T, and here we have two little f's, little f, and then a big T and a little t. So if any of one of these four sperm met this egg, those would be the combinations of the zygote. Well, any one of these sperm could meet this egg, so those combinations would be big F, little f, because this one and this one meets this one and this one, so that would be big T and little t. This would be big F, small t, small t. This would be little f, big T, little t. By the way, you always put the dominant first. Okay, it doesn't really matter if it's on the top or the side here. You always put the dominant allele first. Okay, and then this one's homozygous recessive for everything. Little f, little t, little t. Over here, this would be a big F, big T, big T. This would be a big F, big T, little t. This would be a little f, big T, big T. This would be an F, big T, little t. Big F, little f. Oh, I don't know what happened there. Big F, big T, little t. Big F, T, T. Hopefully these are right. I'm kind of going fast here without thinking. It's Friday and my brain's not working all that great, so if I made any of these wrong, stop me. I guess you can't stop me. You can't stop me. I'm recording this. All right, so anyways, there we go. Hopefully these are all right. But again, it's not anything more tricky than just taking these and adding them to this and putting them here. Well, when you look at, these are all the different combinations that could occur when you have, think about how all of these sperm and all of these egg could be packaged through meiosis regarding these two traits. These are all the different possibilities here. And so when you look, and maybe you're asking the question, what percent of the offspring will have, let's say, purple flowers and maybe wrinkled seeds? Well, if I was asking specifically this question, I'd be looking for anything that's got a big F, because that would be the dominant allele, and then the only way that something could be wrinkled, that's the recessive phenotype, so it would have to have two recessive alleles, because remember, even if there's one dominant allele present, it's going to be the dominant phenotype. So anything with an F and this would be little t's is what I meant to put here, little t's for texture is what we decided up here. Okay, so anything with a big F and also two little t's, that's our criteria. So this has got big t's, so that doesn't fit it, this doesn't fit it, this doesn't fit it, this doesn't fit it. This has two t's but no big F, so that doesn't fit it. This doesn't fit it. Here is one that works. This has got a big F and then two little t's. This has not two little t's, so that doesn't work, that doesn't work, that doesn't work, that doesn't work, that doesn't work. This has got two little t's, but it's got one no big F's, so that doesn't work. This one's got a big T, so that doesn't work. Here's another one that does work, because it's got two little t's with a big F, and this doesn't work. So this one works, and this one works. So that's two possibilities. If this sperm met this egg, we would get that combination, or if this sperm met this egg, we would get that combination. So that is 2 out of 16 possibilities. So when you consider that probability, you would say what percentage of the offspring will have purple flowers or be wrinkled? That would be 2 of 16, which is the same thing as 1/8, which I think is 0.1357, maybe, I want to say. 0.1357. I don't have a calculator on me, but it should be close to it. All right. So about 10 to 15% of the offspring are going to have purple flowers and be wrinkled. Okay. So that's how a dihybrid cross works. The last thing that we'll talk about then is an X-linked trait, and this is going to be inherited a little bit differently, and so require kind of some additional attention to detail. And this is because of the fact that the male-female anatomy is produced by an X chromosome with a Y chromosome on the what are called the sex chromosomes. The female anatomy is going to be designated by big X, big X. Okay, so let's say that there's some mutation on the X chromosome that causes color blindness. And this is kind of a real, I think, scenario, actually. I don't remember exactly the specifics, but I think this is a real scenario. And so if we're looking at our, let's say that the dominant allele would be no mutation. In other words, not colorblind. And then let's say that the recessive allele would be a mutation. And so we would say that that means that it is color blind. Okay. Well, then if we put our allele key together, it looks something like this. We can maybe say that X, and let's say, I forgot to mention this, because this is an X-linked trait, let's say that this is a mutation that's only on the X chromosome. So this is on X, the information that codes for all of this is on the X chromosome of the sex chromosomes. So if it was X without a mutation, and sometimes the way that this is written is a little plus little plus sign, it stands for wild type, meaning if you were just to get it from the wild and it had no mutations, that. And then let's designate, let's say X superscript C for colorblind means that the mutation is present. All right, so let's imagine a Punnett square now, where let's say that there was a woman and a man who loved each other very much and decided they wanted to make some babies, okay? And let's say that the man did not have color blindness, so he has, let's say that he is X, Y. So X plus means no mutation, and then XC means colorblind. So he has the X chromosome without the mutation, and then he has the Y. Well, the Y doesn't really matter in this scenario. It's just kind of there to keep in mind of. Okay, and let's say that the woman is heterozygous. So she has one copy of the X chromosome that has no mutation, but her other copy of the chromosome, the X chromosome, has color blindness. Remember, women are going to have 2 copies of the X chromosome, whereas men only have 1. Well, the gametes that she could put forth, she could have an egg that gets this copy of the X, or she could have an egg that gets this copy of the X, the one that's got the mutation on it. And then the male, they could produce sperm that has the X on it, or the sperm that has the Y on it. Okay. So here, if this egg and this sperm met, it would be a zygote that's got 2 kind of normal X chromosomes on it. They don't have the colorblind mutation. If this egg met this sperm, then there would be a heterozygous individual, so it would be X plus and XC, so they inherited this X chromosome from dad and this X chromosome from mom. Okay, and then if these 2 gametes met, this egg and this sperm, that would be an X normal chromosome with a Y, and then this would be an X with the mutation, the colorblind mutation, and a Y as well. Okay. Now when you're looking at this, you can kind of see why this is what happens when you're talking about X and Y's. You can see that the reason why approximately half the people in the world have female anatomy and half the people in the world have male reproductive anatomy is because of this reason. You know, you can see that 50% of these offspring have Y's, so that's going to lead to the growth and development of male anatomy, and then 50% of them have 2 X's, which is going to lead to the growth of female anatomy. But above and beyond that, you can see that men kind of get the shaft in these types of circumstances, right? Because look at here, the 2 girls that are born, if or the probability, the girls that are born here, if they are born, none of them are able to have just the colorblind allele, or I shouldn't say it that way, but none of them have the color blindness, because if this combination of gametes met, it would produce a baby girl that has both copies of the normal chromosome. If this baby girl was born because this gamete met this gamete, then that baby girl would have no color blindness either, because she still has 1 copy of the normal chromosome. She does have 1 copy of the colorblind chromosome, however, because that's the recessive allele, you would actually need just recessive alleles and recessive alleles alone to have that recessive phenotype. So we would say that she is a carrier of the recessive allele, but she does not actually have that phenotype, because the dominant one here is the one that takes over and is expressed. If we look at the boys that could be born from these combinations, if this egg met this sperm, then that would lead to a baby boy, because he gets the Y chromosome and has 1 normal X chromosome, so no color blindness in here, this boy. But this boy here, if this X met this Y, then it would be a colorblind boy. And because of this very nature of X's and Y's, generally men oftentimes will inherit X-linked mutations or X-linked disorders more frequently than women. So for example, like muscular dystrophy is a disorder that is X-linked recessive, and that is going to be much more common in boys. And there's quite a few others as well. Okay. But in either case, that's how X-linked traits work. And again, you can see it's a little bit different due to just the fact of this Y here, and how men are a little bit more susceptible to getting those because they don't have a normal, they don't have 2 copies of the X chromosome to consider. All right, let's go ahead and do some review. I got some questions for you. Here's the scenario, here's the allele key. We're talking about 2 traits now. We're talking about seed texture, and we're talking about seed color. Okay, and the allele key we have is a big R means the dominant phenotype, which would be round. A little r means the wrinkled phenotype, which would be recessive. And then for the seed color trait, a big Y would mean that the seed is yellow, and a little y would mean that the seed is green. So consider this. We have 2 plants, plant A and plant B. Plant A is heterozygous for the one trait and homozygous recessive for the other trait. Plant B is heterozygous for the one trait and heterozygous for the second trait. Well, if you were to cross these together, I have a couple of questions. First, what are the phenotypes of plant A and B? And then second, what ratio or percentage of the offspring from this cross could one expect to have round green seeds? Go ahead and pause the video and work through this pretty long question, and then see if you're right. I'll tell you what the answer here is in just a minute. Okay, question number 1, when we're talking about the phenotypes again is just the appearance of the plant. So, you know, this plant here, plant A, might have the genotype that's heterozygous. It might have one allele for the dominant, a dominant allele and one recessive allele, but when we're talking about the phenotype, we're only concerned about its appearance. And so plant A, because it has the dominant allele here for seed texture, it will have that dominant phenotype. So it's going to have round seeds to it. And then because it's homozygous recessive, it doesn't have any dominant alleles of seed color. So it's going to be the recessive phenotype, which is green. So these are going to be round green seeds in these plants. And then in this plant, this is going to be round as well because we still have that dominant allele. But now we actually have a dominant allele in the second trait, seed color, so these are going to be round yellow seeds in these plants. Okay, so that would be the answer to question number 1. And then question number 2 is the big one. Okay, so in question number 2 we have a plant that is big R, little r, and little y. This is plant A. And then plant B is big R, big Y, little y. We're going to cross these 2 together. So this is 2 traits, so it's a dihybrid cross. So that means we're making a 4x4 table. 4 columns, 4 rows. Okay, and then we have to figure out what the gamete possibilities are. So the first one would be this meets this, so that gamete would be big R, little y. This would be little r, little y, so put a little r, little y here. Oh, sorry, that's usually not the order that it's done. All right, so then for part so for question 2, we have 2 plants, A and B. B is going to be, or A is going to be big R, little y, little y. B is going to be big R, big Y, little y. And then when we're doing a cross concerning 2 traits, we need to make our big 4x4 Punnett square. like so. Okay. So the types of gametes that plant A could make, it could package this allele with this allele in a gamete, and that gamete would look like this, have bearing these alleles. Or a gamete could have this allele packaged with this allele, and that would appear like this, which is actually the same as that. The gamete could package this allele with this allele, so that would be a little r and a little y. Or it could package this allele with this allele, which would be little r, little y as well. Okay. This one we could start packaging this one and this one up. That would be a gamete that's bearing these alleles. This one could be packaged up with this little y, so that would be big R, little y. Then this one could go with this one, which would be a little r, little y. And then adding these two together would be, this one and this one and this one would be little r, big Y. My mistake. See, I'm telling you, it's Friday. At least it is today, anyhow. Okay, little r and a little y. There we go. Okay, so if we add all of these combinations, we consider all of these combinations, here's the different zygotes that could potentially be made. And this allele key is kind of silly because I can't really determine what's a little y and what's a big R, big Y. So you might think of that when you do a question like this, maybe come up with an allele key that's not so ambiguous. But I'm pretty sure that, yeah, all of these are, I think all of these are little y's. Alright, so I think if I did everything correctly, again, I kind of went fast there, and it's hard to tell if I'm talking about big Y's or little y's here just because my key is a little bit goofy. If I did this right, this would be all the different combinations of offspring that could be produced. And the original question was wondering about having round and green seeds, so we would need there to be a big R and two little y's in order for that to be the case. So we're looking for a big R and also two little y's. And that's not this one, it's not this one, it's not this one, it's not this one. All of these have little y's, so that's good. And all of these have at least one big R, so that would be okay. None of these have homozygous recessive. And then these are all homozygous recessive. But of these ones don't have a big R, so we'll cross those out. So it's basically this one, this one, this one, this one, this one, and this one. So we would expect if these two plants made some little baby plants, right, these would be about 6 of 16 of them would have round and yellow seeds. So it would be whatever percentage that is. It's close to a third, like 37% about of all of the offspring you would expect would have round yellow seeds. Okay, I think that's where we'll end for now. A little bit about Mendel and what he found out and how we use that to, do a little bit of studying about inheritance.